All right, so welcome back. Today I'll be going over two different phylum or phyla. So I'll be going over the phylum Cnidaria, which are jellyfish, medusas, and sea anemones, and I'll also be going over the phylum Platyhelminthes, which are flatworms such as planaria or uh, tapeworms and flukes. So one thing that both these organisms share in common is that they're both symmetrical in some way. Our previous phylum, the phylum Porphyra, didn't have any symmetry. When you look at its body plan, a sponge doesn't, you can't cut it across an axis, no matter how many times you try. The two sides, or how many sides you cut, won't look alike. So in our phylum Cnidaria, or the jellyfish, they have radial symmetry, meaning you can cut it along any axis, and it'll look alike. There'll be a distinctive left and right side. And then our second phylum, the phylum Platyhelminthes, displays bilateral symmetry, so you can cut it across one axis only, leaving a distinctive right and left side. Just like with this blue Morpho butterfly, you can only cut it one way. You can't cut it horizontally, only vertically like this. So everything that we talk about from here on out the rest of the semester will be in this clade Eumetazoa. Our last phylum, Porphyra, is not included in this. So if we're starting with the phylum Cnidaria, and then all the way to the end of the semester will be a member of Eumetazoa. Phylum Porphyra doesn't have tissues, so this is the outgroup for this clade. I keep mentioning the phylum Cnidaria, and so this is a relatively diverse group. It's got 11,000 some species, described species, the majority of which are marine. There are very few freshwater species, but we'll go over three classes: class Hydrozoa, Schistosomiasis, and Anthozoa. So Hydrozoa is stuff like Portuguese man of wars, Hydra, which we'll see in lab, and then the class Schistozoa includes jellyfish. The class Anthozoa includes corals. So a really basic breakdown of this phylum. I already mentioned that they're mostly marine. There's very few freshwater species. They have two stages. So they're polymorphic. The polyp stage is sessile. It's planted firmly into the substrate and it doesn't move. The second life stage that the phylum Cnidaria has is called a medusa stage. And so this is what you would see as a jellyfish or a Portuguese man of war. Those are the modal organisms. They're flowing along in the current. There's two levels of tissue. There's endoderm and ectoderm. They're both separated by this line right here. That's called mesoglea, and that's an acellular, so a non-living structure there. We still have a hydrostatic skeleton. So our last phylum Porphyra, also had a hydrostatic skeleton. So the water pressure of the water column around it is what's giving this organism its structure. Everything in this group, besides class Anthozoa, has what are called cnidocytes. So these are specialized cells that these jellyfish or corals, or not corals, that these jellyfish or hydra use to capture prey. So it takes what's called a nematocyst, which is this barbed structure, and it'll shoot it out at any food items that come in, or prey, or predators that come across this organism. So there's a neat video right here that shows the nematocyst firing. So see they all fire simultaneously. Yeah, there they go. So if you ever get stung by a jellyfish or step on a sea anemone, this is what's happening. They're shooting these barbs, these nematocysts, into your skin. So in the case of some jellyfish, those nematocysts are poisonous. So that is what a nematocist looks like when it fires. When they're not firing, they're just in these capsules right here, and they just wait for some sort of nervous impulse to send those filaments out. So we have our first gastrovascular cavity. So in our last phylum Peripheral, we had intracellular digestion, meaning that the organisms could only eat food particles that were as small as its cells. Now we have a distinctive structure for eating food. So now we can have organisms eating things that are larger than their cells, gaining more nutrients, and then growing larger. When I say this is an incomplete digestive system, I mean that there's only one opening to that digestive tract. There's a mouth, and that mouth also acts as the anus. So we have our first nervous system. It's called a nerve net. So if you look at the bottom picture, you can see there's no central brain or condensing of the nerves, but there's this nervous system. So these organisms are sensing their environment. If need to, if they come across a prey item or they're being attacked, they can use that nerve net to stimulate those cnidocytes and fire off the nematocysts. They reproduce sexually, so there's both males and female individuals, but they can also reproduce asexually through budding, which you'll see on Wednesday or Thursday, or via fragmentation. So if you break off a piece of coral, it can create a new individual. So one of the things that is neat about this phylum is that they display what's called alternation of generations. So you can have one stage being these polyps, so those sessile individuals that are on the ground, they're not moving. Then you can have in that same generation these modal medusa stages. So you can have the jellyfish or the Portuguese man of war. And so every so often, we'll go through these alternations. So our first class, so we have the phylum Cnidaria. This is our first class, Schistosomiasis, and these are the true jellyfish. The way we define these classes is that we use the dominant stage of their life cycle. So in this class, the dominant stage is the medusa stage. They spend very little time in the polyp stage. Our second class is dominated by the polyp stage. So this includes Hydra, which you'll see in lab Wednesday or Thursday. Also includes Portuguese man of wars like this one. So this is the medusa stage. This is what Portuguese man of war looks like much more frequently. It's really only in the medusa stage for a very short time. So if you see a Portuguese man of war, just know that it doesn't always look like that. There is an invasive species in this class. It's found basically almost in every slow-moving freshwater area in the world. It's probably one of the most invasive species out there. So it's native to the Yangtze River in China, but as humans like to do, we transport things around by boat. A couple of those polyps ride, and then they spread throughout the world. And then we, as Americans, spread it even more by stocking fish. So the polyps are microscopic. It's hard to control. And as we stocked fish in the early 19th or early 20th century, we also transported this hydrozoan across many miles of rivers and lakes. So here you can see it's predominantly in the eastern half of the U.S. It is found in Kentucky and Tennessee, but there are some spots such as in California and Idaho, Nevada. So the anatomy of a hydra, we'll go over this in lab in much more detail, but just know that it has a mouth at the very top. It's just a single opening, so that mouth acts as an anus and a mouth. It has tentacles, which it uses to grab prey, and then it uses hydrostatic pressure, so pressure the liquids inside of it to bring those tentacles and the food closer to its mouth. It has a stalk, which is this entire structure here. Basically, that's the outer layer covering the gastrovascular cavity. And then it has the basal disc, which is this point right here where it attaches to the substrate. If we look at the internal anatomy, you can see it's relatively simple. There's not much going on. So you have the ectoderm and the endoderm right here. And then you have this thin line that separates the two. That is the mesoglia. Within the ectoderm, you have these cnidocytes, or those stinging cells. And then within the endoderm, you have the gastrovascular cavity. So that's where it's digesting food. So yeah. The third class in this phylum is the class Anthozoa, and these are the corals and sea anemones. And so this class is dominated by the polyp stage. There's no medusa stage, so they're totally sessile. Well, not totally. I'll show you a video in a little bit. One of the ways they reproduce is through petal laceration. So as they move along the seafloor, they can get these small cuts along their basal plate, and as that happens, small individuals are cut off, and so those individuals grow into new adults. A couple of them, such as this bright green one right here, have the mutualistic relationship or symbiotic relationship with photosynthetic dinoflagellates. So those dinoflagellates are producing glucose and such, and so it's meeting the Anthozoa or the sea anemone's needs, and then the sea anemone is providing protection. So here's a neat little video of a sea anemone moving. It's moving along the base of a reef tank. And so in the course of 30 seconds, in this time lapse, it moves quite a bit. So I believe this was taken over an hour. So, but as it's moving along, it's seeing small scrapes on its basal plate. And as that occurs, new individuals are being cut off. So it's reproducing via petal laceration. One of the biggest threats to class Anthozoa, and really to a bunch of other organisms, is ocean acidification. So as we come into larger or warmer temperatures, much more CO2 dissolves into the oceans. So that increases the acidity of the oceans. So most of the invertebrates, including the class Anthozoa, use calcium carbonate in their shells or in their skeletons. And so as the oceans become more acidic, their the amount of calcium carbonate in their shells becomes much lower, becomes thin, and they become susceptible to more predation and such. The increasing amount of CO2 in the water also lowers the amount of dissolved oxygen, and so every organism in the ocean needs some level of oxygen for it to metabolize. And so as there's less oxygen molecules in the water, those organisms tend to die off. So there's a neat study published in 2014 that measured the decrease in shell thickness in some invertebrates. And so there's a British expedition during the 1870s that collected these small crustaceans. As they preserved them. So in 2014, there was another expedition that collected these invertebrates, and then they measured the difference in shell thickness. So as you can see, in the much more the older invertebrates, they have a shell thickness of around 25 micrometers. The one from 2013 or 2009 has a thickness of around, I'm guessing that's 10, so of around 10 micrometers. So that's really a 70% drop, close to a 70% drop in shell thickness. And so you can see this, they present it a couple different ways. You can see along the inside of the shell, the thickness is decreasing. You can see that here. These blue lines represent shell thickness. So increasing ocean acidification is measurable, and it's been occurring for hundreds of years. So for those of you that use the chart, this is what you would fill in. So we just went over the phylum Cnidaria, and in lab we're going to go see Hydra. So it's in the class Hydrozoa. All these organisms are aquatic. There's no terrestrial cnidarians. And for the most part, they're all marine. There are a few exceptions, such as Craspaduca sowerby, the invasive hydrozoan from China. They're diploblastic, meaning they have two layers of tissues, endoderm and ectoderm, and they display radial symmetry. For their skeletal system, they have a hydrostatic skeleton or hydrostatic system, so they use water pressure within their body, and they increase the pressure compared to the water column around them. They reproduce one of three ways. They can bud, like this individual right here is doing. So this right here is a bud, and that will break off and become a new individual. They can reproduce via fragmentation. So if you break off a piece of the coral, that will grow into a new individual. They can also reproduce sexually, so they produce both male and female. Well, they have male and female individuals. They have an incomplete digestive system, so there's only one opening to that digestive system, but they have extracellular digestion. So in our last phylum, Porifera, we had intracellular digestion. They were only eating food particles that were small enough to enter the cells. Now we have organisms that have extracellular digestion, meaning that they're eating larger bits of food. They have a nerve net, so they don't have a central nervous system like we do. We have a brain. They have their nervous system spread out throughout their body. And for the most part, well, they're both modal and sessile. The next group that we're going over is Platyhelminthes. So we're skipping 3 different phylum. We're skipping actually a lot, but we don't have time to fit them in. So we're skipping the phylum Ctenophora, the phylum Xylomorpha, and the phylum Rotifera. So we're going to jump right into Platyhelminthes. First, everything from Rotifera to Arthropoda is what's called a protostome. So a protostome is what we consider organisms where the developing bundle of cells, or the morula, the first opening to that bundle of cells is the anus, is the mouth. We consider deuterostomes, such as echinodermata or chordates. So humans are deuterostomes. So the first opening to that bundle of cells is the anus. But for now, these next several phyla that we cover are in the are protostomes. So the first opening to that bundle of cells is the mouth. Also, the next couple of groups that we go over will have this bundle of cells. They're considered Lophodrochozoans. So at some point in their life stage, they'll have, in their larvae, they'll have this ciliated band around their body and then around their mouth. So within those 3 clades, so we had the clade Eumetazoa that was on, I believe, the second slide. We had the clade protostomes, and then we had the clade Lophodrochozoa. So within those 3 groups, we now have our third phylum that we've covered in this class, or will cover in this class, which is Platyhelminthes. And so literally, Platyhelminthes means flatworm, and they are exactly that. They are mostly aquatic. There are very few free-living or terrestrial species, and for the most part, they are parasitic. A couple of you were lucky enough to see some of these parasites in the tapeworm, floating about, living in that tapeworm's body. So there are free-living individuals. They're not all parasitic. They're triploblastic, meaning they have 3 layers of tissue. They have mesoderm, endoderm, and ectoderm. We'll go over that in a little bit. We now have our first group that's displaying bilateral symmetry. So it has a central axis, which you can cut its body down. And for the first time, we have cephalization, which is extremely important. So if you have an organism that's chasing after a prey item, it's nice to have some sort of central area where it has this nervous system. And so we consider this cephalization, basically the formation of an anterior head. So in all of our classes, you have this anterior area where it has a central nervous system. And in our planaria, it has eye spots. So we have our individuals reacting to different light, different amounts of light in their environment. And so they use this to find food. We have gastrovascular cavity still. So this individual is going to have an incomplete digestive system. It has one opening on the bottom side of it. So right here is its pharynx, and then right here would be its mouth and anus. So I just went over the nervous system a little bit. I'll go over the excretory system in a little bit more detail. I'll go over all these in more detail, but in a little bit. So our first class is class Riberia. So these are the planaria. This is what you'll see in the lab Wednesday or Thursday. These are free living. There are no parasitic individuals in this class. In class, you should be able to see a head, and you will definitely see eye spots. So these areas where that individual can react to different amounts of light. It has both a mouth and an anus on the bottom side of its body. Its pharynx is the direct connection from that opening to its digestive system. We'll also look at cross sections of these individuals while we're in lab. And so you'll see the three different layers that this organism has. So you have ectoderm, that outer layer. You have mesoderm, which is all this stuff right here and here. And then you have endoderm in the middle. We also have gastrovascular cavity, where this organism is digesting food. So we consider these individuals acoelomate, meaning they don't have a body cavity. So we are coelomates, humans are, and all other chordates are coelomates. So we have this open area inside of our body where our intestines are. Everything in the phylum Platyhelminthes is an acoelomate, so it lacks a body cavity. So for the digestive system, I mentioned that it's incomplete. So there's only this one opening to that system. It's branched out like this, basically because it doesn't have a true circulatory system. So in order to get nutrients to all parts of the body, it just has this really long gastrovascular cavity. And then the branching structure also increases surface area. So the more folds you have in an area, the more surface area, the more food particles you can digest. It's got a neat nervous system. So it's got these transverse nerves that run across its body, and then it has these parallel nerve cords that run the length of its body, and then it has this centralized area called neural ganglia. So those neural ganglia are right underneath those eye spots. So it's reacting to the environment. So it also it really resembles the nerve net found in the phylum Cnidaria, but this is just another step in evolution. So these individuals reproduce asexually, so they produce both gametes, male and female. So you'll have both the testes and the ovaries in a single individual. For the excretory system, we now have individuals that are not just using diffusion, they're actually pumping waste out of their body. And so in the phylum Platyhelminthes, they use protonephridia or flame cells. And so this is connected to the digestive system. So our second class is the class Trematoda. These are parasitic. So the most infamous is Schistosomiasis. So this is a freshwater trematode. And so it has a pretty complex life cycle. It infects humans, snails, and I think there's a second or third host in there as well. So you have distinctive males and females. So if you look at this SEM photo, you have this male right here, and then you have the female sticking her head out right there. So they have pretty complex life cycles. If you look at this one right here, Riberia odontre, it has a fairly complex life cycle involving 3 or 4 hosts. And so this is a pretty neat, it's actually ongoing problem. So in the early 1970s, people were finding these amphibians with lots of deformities. So what they found was that this trematode was laying cysts in the joints of these amphibians. And so amphibians, as they were developing, they would grow a second or a third limb, or the limb would just be stunted. So you can see that the stunting of growth in this frog, and then you can see the multiple legs here in this frog. So the life cycle, it begins in snails, and so the snails pass on that trematode to tadpoles. From there, those trematodes grow in the cysts of the legs as they're developing. This results in indirect mortality of the frog. When you're carrying around an extra limb, you can't jump as far, you can't run as fast. So those individuals are picked off by birds, and so trematodes are being passed directly from the frog to the bird. From there, the trematodes reproduce, and then the life cycle starts over again as the trematodes are passed from the body into any freshwater source. So this is what it looks like under SEM. You can see it's got this sucker here, and this is what the larval, the cyst consists of. So in each of those cysts in the amphibian's limbs, there's one of these individuals. And so this gets passed from frog to birds, and then from birds to snails. So our third class in the phylum Platyhelminthes is the class Cestota. Once again, these are entirely parasitic, and these are the tapeworms. So if you've ever had your cat or dog with tapeworms, you know that they are pretty nasty organisms. So because they're so parasitic, they're so well defined as parasites, they've been gradually selected against having a digestive tract. So when you're just living in the intestine of another organism and you're just absorbing nutrients, you don't need to have a complex digestive tract to break down any food particles. So they lack this digestive tract and they just use absorption as their method to gain nutrients. They have a scolex, which I'll show you in the next photo. Their bodies are made up of these structures called proglottids. And so each of these, well, this is a better photo here. Each of these structures here are proglottid, and there's different maturities. So you can have the immature, which lacks any genitalia. You can have mature, which will have both testes and uterus, and then you can have the gravid proglottids. And so gravid proglottids are filled with eggs. And you should be able to see that in lab on Wednesday or Thursday. Inside of each of these proglottids, you have both the testes and the uterus, or the ovaries. Right here, this is an ovary. Right here, that's the testes. Those produce both sperm and eggs. The eggs travel down the uterus and then out of the genital pore. It uses Filaria to create yolk, basically some sort of nutrition for the developing eggs. So here's a pretty good SEM photo of a tapeworm, class Cestota. So up here, you have the scolex, which is just this large structure here. This tip right here has these barbs, and so that's called the rostellum. And so it uses those barbs to attach itself to the inside of the intestine. And so it uses the suckers then to absorb nutrients. And then you can see each of these segments here, each of those proglottids. So those of you that use the chart, we're talking about the phylum Platyhelminthes, and then in lab you'll see examples of Planaria, from the class Turbellaria, and you'll see a slide on class Cnidaria, so the tapeworms. For habitat, they're completely aquatic. For symmetry, they display bilateral symmetry, so you can only cut this individual in half, so there will be a distinctive left and right side. It's triploblastic, has endoderm, ectoderm, and mesoderm. It's not segmented. It still has a hydrostatic skeleton, so it has an increased water pressure inside of its body compared to outside of its body, and so that's what gives it its shape. It can reproduce through fragmentation, and then it produces both male and female gametes. So an individual will have ovaries, and it will also have testes as well. They have an incomplete digestive system. Their mouth is right on the bottom of that individual, and it acts as an anus as well. It's the only opening to that digestive tract. It doesn't have a circulatory system, so that's why it has that branched digestive tract, and it has those protonephridia spread out throughout its body. So it can't circulate nutrients throughout its body, and it can't circulate waste away from its body. So it has that branched digestive tract to get food particles as close as they can to each cell. It also has those protonephridia, the flame cells, spread throughout its body so that cells can release its waste. It has a ladder-like nerve system, so it has those transverse nerves that run across it, and it has those two nerve cords that run the length of the body. It has a central neural ganglia right here, which you can consider as its brain. And then it has these two eye spots that it's using to sense different light or sense the amounts of light in its environment. So they are modal. They do move around. You'll see on Wednesday or Thursday that they move rather quickly too. Next week is our osmoregulation lab. So we'll be testing basically different salinities on the weights of goldfish.